The next speaker is Jorge Plutzky, who's the Director of Preventive Cardiology at Brigham and Women's Hospital, and who's going to tell us about some of his work to use epigenetics to understand cardiometabolic disease and hopefully to treat it. Jorge.
Thank you, Calum. I appreciate the opportunity to present here and discuss some of our approaches around this. The clustering of abnormalities one sees in cardiometabolic disorders includes conditions that we might predict to be present, like diabetes, but also others that we wouldn't a priori expect to travel and associate so tightly with cardiometabolic issues, problems like sleep apnea and Alzheimer's or cancer. At the same time that this clustering suggests that there are proximal mechanisms that must underlie why these associate, it's also true that as one moves towards more physiologic conditions and improves energy balance or maintains it if you've never lost it, the improvement in so many of these parameters also supports this notion that there are fundamental pathways that govern this relationship of energy balance and complex states of both health and disease. And I would like to raise with you a conceptual platform of how we've approached this that we think has therapeutic opportunities. And that's an underutilized, underexploited approach to issues around therapeutics, which is to harness endogenous physiology that protects against these problems or that have gone wrong in the development of these issues, and use those to identify modalities in which one might treat, either through a pathway or by having identified specific mediators that might serve as better templates for therapies. So I'm going to provide a couple examples of this conceptual platform and how we've advanced along those lines. And one way that you could explain why these complex phenotypes aggregate together is through the action of key proximal nuclear receptors that, when activated by their ligands, govern not just one target gene, but many target genes, because they convert from being a nuclear receptor to being a transcription factor. And the fact is that the PPARs and the retinoid receptors are critical players in determining transcription of many genes involved in energy balance. But the endogenous ligands and the pathways that generate those ligands have remained relatively obscure until recently. And this is particularly appealing because we know that these receptors are druggable targets, but yet we've operated really in the blind, not knowing what their physiologic partners are or might be. And if we were able to have that insight, we could arguably come up with better therapeutics with templates that might have greater efficacy and safety because they're mimicking and complementing and replacing what's been missing in someone or has gone wrong as part of why the problem arose in the first place. So two examples of how this pursuit of proximal mechanisms that govern these complex phenotypes I want to show you. One is that the interaction of different specific lipid substrates with different distinct lipases can generate endogenous PPAR alpha ligands. And just like they do in endothelial cells, this pathway can potently limit other models of inflammation, as we see here with dermal inflammation in these cross sections of mouse ears, where we can block the very potent inflammatory effects of certain model stimuli if we have leveraged known pathways of lipase and lipoprotein interactions. And this raises a therapeutic possibility that we're actively pursuing, that one could engineer this pathway by designing substrates in order to harness this potential and do so in a way that's much more not just efficacious, but much safer as well. And here's a second example. When we apply these same approaches to retinoid receptors, which are partners for those PPARs, and look at how those endogenous mechanisms of ligand generation occur here, we've uncovered really surprising effects that by modulating these pathways of ligand generation, we can protect mice against many of the effects of diet-induced obesity, keeping them from developing diabetes, for example, and are doing so by activating the white fat to turn into a brown fat phenotype, wasting energy that the mouse doesn't need and thus protecting it, and decreasing issues like obesity, diabetes, and fat accumulation in the liver, all by having gained this insight into how those ligands are being generated. And this creates therapeutic opportunities in mimetics for this pathway, as well as inhibitors that can, again, harness the insight into the endogenous mechanism, but now bringing that to a therapeutic approach. Yeah. Now, as someone who spent really years studying transcription factors and how they interact with DNA, it's fascinating to now have a whole new chapter, a whole new perspective open up in a completely different level, a completely different stratum, organizing principle of what can control these pathways, and that's through epigenetics. And epigenetics refers to the fact that we have a solution to the physical challenge of how one handles literally miles of DNA and can compact it into the nucleus. And we do that by wrapping that DNA around spools known as histones. And then those tightly wrapped coils of DNA around the histones have to be opened up in order to allow transcription factors to gain access to that DNA. And the epigenetic code, as opposed to the DNA code, is completely independent of the DNA, but has similar complexities. And one way of summarizing that is that the marks on these histones are placed by certain proteins known as writers. They're removed by another group of proteins known as erasers that take away those marks. But there's a third family that we've been particularly interested in, which are histone readers, proteins that come along and read those marks and allow for assembly of this transcriptional complex to occur and become another way of organizing these programs. And in particular, one of those are these BET-bromone domain containing epigenetic reader proteins, BRD2, 3, and 4. Now, the BETs have received tremendous attention in the oncology world because when you look at very complex phenotypes like myeloma, it turns out that the BETs are acting at certain specific enhancers to govern that very complex program and reduce it to a much more tractable group of targets. As outlined here, looking at what super enhancer regions contain these BETs, in particular BRD4, and that these accumulate in a subset of given targets. And that this accumulation at certain enhancers has raised this term of super enhancers. And we've brought that to consider issues in inflammation and atherosclerosis, and I won't be talking about work looking at that in adipogenesis. And so here's the results that we find when we interrogate this system in terms of atherosclerosis. And so this is looking at the accumulation of BRD4 signal at different super enhancer elements and the genes associated with them. And down here at the bottom, you see in the absence and then presence of an inflammatory stimulus, the mountains of BRD4 accumulating upstream of this canonical mediator of inflammation and atherosclerosis, VCAM1. And in fact, when you do this across the genome, the whole genetic landscape, you can account for key aspects of the components of atherosclerosis we know exist. And coincident with that, when one treats mice prone for atherosclerosis with an inhibitor of this BET pathway, you can protect those mice from developing atherosclerosis. And this new window into what organizes these programs has many potential therapeutic opportunities. The first is through further development of BET inhibitors with differing selectivity and specificity, how they're delivered, and how we might use them. And the second one is that now that we know that this exists and accounts for this program in a completely new way, would be orthogonal approaches to the same pathway. But the third one, I think, is also compelling and of interest, which is that if we had this technology in 1950 and just simply asked where was BRD4 accumulating, we would have identified multiple targets. We would have leapfrogged the field by decades because it was going to take decades to find those. And I think that's intriguing because when we look across the genome at where BRD4 is accumulating, yes, It's accumulating at a group of 41 proteins of this much more tractable set of 152 that we know are involved in atherosclerosis, validating this whole approach. But we also find novel targets, including a group of 38 proteins that are known but have no known role in atherosclerosis, 31 other targets that are completely new, and an exciting prospect of another 42 that are completely unannotated, that we don't even know what they are, other than they appear to be a functional gene or microRNA. And so hopefully what I've laid out for you is a conceptual platform that one can leverage endogenous mechanisms to identify novel and ideally better therapeutics, and several examples of how that might be done based upon leads we already have and that are offer opportunities for partnership in bringing these leads forward and towards a more therapeutic objective. Thank you.